My name is Or Zook. I'm a faculty member at the Statistics and Data Science Department at the Hebrew University of Jerusalem. And today I'm going to give a tutorial about some generative modeling methods and their application in biology. So the focus will be on the methods, but I will also highlight some applications. And just to note, so most of these methods were developed in the machine learning community or the computer vision community, thinking about applications on imaging. But it turns out that they might be useful for other domains, including computational biology. And I think, I don't know about this audience, how many of you know the details of these methods, but I think it will be very interesting to learn and try to think of interesting problems in biology that you can apply these types of methods to. All right, so let's start. So this is the overview. So I'll give some introduction about what is generative modeling, and then talk about three methods. One is variational autoencoders, then diffusion models, and then a newer method called flow matching. All right. So in generative modeling, our goal is to learn some high-dimensional distribution. It could be biological, it can be something else. And it can have many applications in biology, like highlighted here, but also, of course, in other domains, like imaging. And there are many, many methods, and like I said, we're going to focus on three of them. So before going to the methods, I want to present the standard problems in generative modeling. So basically, we have samples from an unknown distribution. So sometimes it can be just xi's, right? Some samples. Sometimes they can be pairs, xi's and yi's. And these xi's are of high dimension, right? So you can think of images, or maybe the entire gene expression in a cell, or maybe a protein structure, etc. And our goal is to learn the distribution from these samples. And that's the main goal. But what does it mean to learn the distribution? Maybe to evaluate the likelihood, or maybe to have some description of the density function, right? So our focus will be on a slightly different task, which is if I know the distribution, then I should be able to sample a new sample from this distribution, right? So that's like a main focus in generative modeling. So you have your samples, and then you learn a model, and then you want to sample a new one. So that's unconditional generation. A more useful task is conditional generation. So many times I don't just want to sample another example which is like the samples I had in my original sample, but I want to condition on something. And there are also other applications like denoising, imputation, and like I said, likelihood evaluation. All right. So we'll start with the first method, which is called variational autoencoder, but I will frame it in a more general framework of latent variable models. All right, so maybe, I'm sorry about yeah, so I should have removed this slides. All right, so in latent variable models, we have a distribution of X, so that's what we observed, but we assume that there is a latent variable Z, such that we have a joint distribution, p theta of x and z, parameterized by theta, such that the distribution of z is relatively simple and not parameterized. And then we also have the conditional distribution of the data given z. But the issue is that we don't observe z, we just observe x. And why do we use these latent variable models? Because they tend to simplify the distribution. So for example, you can think of distribution of images, let's say handwritten digits in the famous MNIST dataset. So you have the joint distribution over all the pixels of the image, but you also have some latent representations. For example, is a digit 5 or 4 or 6, or maybe is a handwriting, that has a thick pen or an arrow, or is a person who wrote it left-handed or right-handed, etc., etc. So you have this hidden representation, and once you know this hidden representation, you have a much narrower distribution of the observed image, right? So this is a motivation for trying to model these hidden variables. And this is true for almost anything, any interesting signal that you can think of. For example, if you have a single cell expression data, then you can think of the hidden variables like the cell type, or maybe the stage of the cell cycle, or is the cell under stress or not, and things like that. So this is like a compressed representation of our data, such that it doesn't determine necessarily our data deterministically, but the conditional distribution of the data given the compressed representation is narrower. All right, so that's a motivation from latent variable models. And I'm going to skip that, and now I'm going to go to the definition. So in particular, in variational autoencoders, we have this latent, it is a type of latent variable models, and it has, the latent variable has a very simple independent Gaussian distribution. And from this Gaussian distribution, we have a parameterized transformation, p theta of x of the data given z, and we also have the transformation in the other direction. So we want to conditional on the data, we want to ask what is the distribution of the latent variable. So in contrast to simpler models in the past that were used, which some of you are, I guess, heard of, like a mixture of Gaussian models or PCAs, many models can be sort of as latent variable models. Variational autoencoders are much more general, because the transformation from z, the unobserved, the latent variable, to x can be very complex, and it's modeled by a deep neural network. Right? So this makes this model much more powerful in general compared to previous latent variable models. But then we have like the computational problem of fitting these models or training them is much more, it's much harder. So basically the problem is that while we can easily go from z to x, we cannot easily go back from x to z, because we have to integrate over all the possible z's, right? So once we observe x, we don't know this, we have to integrate over the uncertainty on z. So if we want to say maximum the likelihood of the parameters of the neural net, or any other loss you can think of, then this is in general intractable. Right? So one naive solution is to say, okay, well, let's just sample these, sample many samples from the observed, from the latent variable, and then approximate the likelihood by sampling. This, however, doesn't work, because if you have a certain x, you can think of, let's say, an image or an expression vector, then the proportion of latent variables that really are compatible with that is very, very small compared to the huge space of the latent variables, right? So you can, for example, have the wrong cell type or cell state, etc., etc. So basically, most of your samples from Z are useless in terms of approximating this likelihood. So what was the idea of variational autoencoder is to replace this original distribution of Z by some other distribution. It's called an importance distribution. You can view it as a type of idea coming from importance sampling, which is a quite well-known idea in statistics. So you want to sample from a different distribution that is compatible with X, with your observation. And when you estimate your likelihood, you have to correct for it, so you have to divide by it. And so that's the idea of importance sampling, but in variational autoencoder, you don't really know this optimal importance distribution, so you learn it. So what we are doing is we have an additional parameterized family of distributions, also implemented by a neural network, such that we sample we train this distribution, we train the parameters such that they approximate the true conditional distribution of the latent variable given our sample. So now we have, we get to the objective of a variational autoencoder. So we can write the likelihood of our data, the log likelihood of our data, as this complicated term, which has really three terms. So one is the expectation with respect to our Q. The other one is the distance between Q and P of Z, so that's a prior of Z. And there's also another distance here. And by the way, these distances are Kolbe-Kleiber distances, so distances between distributions. And the important thing to note is that this part, the third term, we only care that it is positive, right? We don't try to optimize it. This is a hard one to optimize. So what we are doing instead is we are optimizing the first two terms. And that's what's called the evidence lower bound, or ELBO. So we know it's a lower bound for the true likelihood. And if our neural network for implementing Q has a sufficient capacity, so it's deep enough and wide enough, etc., then we can approximate well this true conditional, and then our approximation will be very good, right? So this term will be very low. So that's the idea behind variational autoencoders. Now there are all some other technical issues that I think I will not get into. So this is the first equation is basically the sample version of the equation of the distribution. And there's also another technical issue is that you cannot really directly sample from Q. You need, because if you directly sample from Q, your samples depend on your parameters that you are trying to estimate. You want to push this parameter to the gradient. So you need to sample from some other distribution that doesn't have your parameters and represent Q as some, in this case, linear transformation of this epsilon. And then once you do this representation, the parameters here that you need to learn go into the gradient. So eventually what you get is you have some optimization problem, and you can compute right? So the optimization problem has two terms. One depends on both phi and theta, the parameters, and the other one only on phi, and you optimize them directly. So you can compute the gradient and then apply standard techniques in machine learning, stochastic gradient descent, or more advanced iterative method like ADAM or momentum gradient, etc., etc. And that's how you train the variational autoencoder. All right. Now let's talk a little bit about applications. So while I talked about the application of sampling new examples, there are also other applications of interest. So I just highlighted a few methods in the single cell expression analysis field, but there are many others. So basically, once you learn a hidden representation of your data, you can look at the hidden representation and use that for many downstream tasks. So, for example, removing technical confounders, clustering, etc., etc. And there are also so this is method just working on single cell RNA-seq data, but there are also methods that work on multimodal data. So, for example, single cell plus protein or chip site signal, or like we heard a couple days ago of spatial transcriptomics together with in situ hybridization data. All right. So I apologize, but I think now I realize that this is the wrong version. That's why it was not showing what I wanted. Yeah, okay. Yeah, sorry, AJ, I opened the non-updated version, so this should be cleaner. And yeah, how do you go to full screen?
I think Command L or Control L.
Control L. Okay, great. Okay, that's much better. Apologize. Okay, so yeah, so now we are on the slide of single cell. So like I said, we have a method that just work on one modality, for example, RNA expression data, or we have multimodal methods that can work jointly on RNA counts and surface protein data, or spatial transcriptomics and histological data. And these multimodal applications are especially relevant for generative modeling because many times you learn your model from samples that have both modalities, but then you can generate conditional samples on new data, on new samples where you you only have data from one technology, and then you can sort of impute the other one. So that's a very, very useful application of generative modeling. All right, so that was a variational autoencoder. Now I want to move to the second framework, which is called diffusion models. So in diffusion models, there are some similarities, but also some very different characteristics compared to variational autoencoders. We still have this data coming from an unknown distribution, and we want to model it. So we want to model it by taking it to be some modification of a simple distribution. But this time we don't work in a latent space. So you see we are in the same space, but we are simplifying the distribution. So basically, if this is our data distribution, for example, in images, this is from the original paper about diffusion models, and we think of like a time series, right? So we have 0, 1, 2, 3 until t, so we have very, very small steps. And so at time 0, we have the data distribution, which is very complicated, but we have samples from it. And at time t, we have a very simple distribution. For example, Gaussian IID, right, for each feature. And now the goal is, in diffusion model, is to learn the path that goes from the complicated distribution to the simple distribution or vice versa, right? So if you go into the details, so the direction from the data distribution to the simple distribution is quite straightforward. You just add Gaussian noise, right? So going from here to here is simple. This Q distribution is just additive Gaussian noise. What's hard is learning the other direction. So this will be implemented by a neural net, right? So basically what we are trying to train here is a denoising neural net. So it gets us, in the training set, it gets pairs of a noisier version of the image or the sample and a less noisy, a cleaner version. And it tries to learn the denoising part. Right? So that's the idea behind diffusion models. And in contrast to variational autoencoder, you see that we have this very, very, a large number of very, very small steps. And this is important. It has a benefit and also a downside. So the benefit is because we are doing very small steps, the approximation we are using here in our neural net is much more accurate, because if you do a local step, it's right. Once you have that xt given xt minus 1 has a Gaussian distribution, if the variance is small enough, it's also correct that xt minus 1 given xt has an approximate variance normal distribution. Whereas in a variational autoencoder, we do a one giant step, and then the approximation is not as good. So that's the advantage of diffusion model, and that's why they are so interesting and popular. The disadvantage is because we have to do so many steps, then the computational burden is much higher. So they are much harder to train computationally. All right. So we have, we have this model of, if this is a noise model, so we just add Gaussian noise, and this is the denoising model, so it's still Gaussian, but notice that the mean and the variance of the clean sample given the noisy sample are some nonlinear complicated functions that are implemented by neural networks. All right. So again, like in the previous example of variational autoencoder, we also can take the log likelihood and derive a variational lower bound on it. So because we have the same structure of going back and forth in the transformation, and then you can train this model using this approach. There's another another issue that does not appear in variational autoencoder is that when we train the data, when we train the model, we take the data and we ourselves apply noise to it. So you take your clean data sets and you sample the the strength of the noise. So you can do small noise or large noise. You sample that uniformly, and so you get in your simulated training set, you get like many denoising problems of different noise strengths, and you train them all together. So that's basically like a pseudocode of the algorithm, and this is a pseudocode of the algorithm for sampling new ones. So once you train your model, it's pretty easy to sample a new sample from your distribution. You start with the noisy sample, which is very easy to sample, and then you just apply the steps. So you go all the T steps of the neural net, and you denoise it in very, very small steps. So while this is straightforward, you can imagine that it's computationally intensive because you have many small steps here. All right, so some applications and results. So these are from the original, this is from a subsequent paper that actually combines the idea of diffusion model and of latent model. So instead of doing the diffusion model on the original X, it first learns a latent parameterization, a latent representation, and does the diffusion only on the latent representation. And I don't know if you can see it, but it significantly improves the resolution of the images using this latent representation. And another line of methods that you probably all know of, maybe play with, is combining this diffusion models with transformer models that are used for text analysis. And then you can have multimodal models that model jointly images and text, and you can use them, for example, to do conditional generation. So given a description of an image, you can generate an image that corresponds to this description. All right. So now what about biological applications? So I think the most important application is for protein design. So I'll try to explain why that. And there's a very influential paper called RF diffusion. And the goal in protein design, there are many problems in protein design. One of them is to design new proteins that do not exist in nature, but they have structures, 3D structures that are similar to the distribution of structures observed in nature. So you can take all the known proteins with known structure, for example, from the protein data bank, PDB, and you can use them to train a model, and then you can sample from this model new proteins. And of course, in this application, you also need to sample the sequence, so the model is multimodal because you have the joint distribution of the sequence and the structure. But the idea is very similar, and they actually use diffusion models. So you start with a Gaussian distribution over the structures, and then you apply the symbol steps until you get a protein structure, which in many cases looks like very similar to known protein structures. So that's sort of the analogy between the images space to the protein space. And so how did they do it? So that's also, I think, an important point to note. The architecture is very complicated. It's called the Rosetta-fold architecture. It's similar, for those of you who know, to AlphaFold architecture, which was developed originally for a supervised learning task. So for predicting the 3D structure from the protein sequence. But once you have this architecture, you can use it as a denoising architecture. So you can insert a noisy 3D structure and let the architecture denoise it. And once you have that, you can plug it into the diffusion framework. So it's a very nice idea. So from a neural net that is able to solve a supervised learning task, you can now retool it to do design, so unsupervised. And these are some examples of the results. You can do unconditional design, just generate new proteins, or you can do some conditional designs. For example, you can force symmetry on the protein, or you can say, I have this other target and I want to design a protein that binds it, etc., or have some functional motif, etc., etc. So these are all examples of things that you can do with these architectures. All right. How am I doing on time? Good. All right, good. So now, so that was different. Now let's move to the third method, which is called flow matching. So before that, I will just highlight again the benefits and limitations of diffusion models. So they have a much higher capacity compared to variational autoencoders, and they reach both in protein world and also in images a much more accurate results, but they are very, very computationally intensive. And so the third method I'm going to describe, which is quite recent from 2022, it's called flow matching. And the idea there is to do something that is similar to diffusion, but is simpler. So the goal is to get the benefits of the diffusion models, but with less computational burden, right? So I will not go into the details of flow matching. I'll just highlight the major ideas and differences from diffusion models. So in diffusion models, we go from the simple distribution here to the complicated data distribution, and we go in a stochastic manner, right? So we sample from the simple distribution, and then we add noise, add noise, add noise. So it's a stochastic process. In flow matching, we go from the simple distribution to the complicated distribution via deterministic flow, right? So you have a randomness when you sample your initial sample from the simple distribution, but once you sampled it, it determines your point in the complicated distribution deterministically, right? So that's a simplification, right? So it's deterministic. On the other hand, it's more general in the sense that there are many different ways to go from here to here, and I will not go into the details, whereas in diffusion there is only really one way to go from the simple distribution to the complicated. There are many, many different flows that will go from the same distribution here to the same distribution here. And by the way, there's also, I think yesterday someone mentioned optimal transport. So one of the ways to train these models is via optimal transport, which is trying to find the shortest flow that will go from here to here. Another important idea of flow matching is this velocity field. So if your flow, which we parameterize by psi of t, goes from here to here, then you can think of its derivative, which is basically telling you at each time here, how are you going to change? How are you going to change your distribution? That's what's called a velocity field. That's this u. So it's a vector field that we have here and here, here. So it's a vector field that is dependent on time. For each time point, we have a different vector field. And instead of training a neural network that will try to predict this transformation, it turns out that it's simpler to train a neural network that will try to predict this u, right? So this is an important computational simplification that was introduced in this flow matching. So we are trying basically to learn the vector field u and not psi. And the way we're doing that is we are actually doing conditional vector field. So instead, we are not trying to learn in the distribution here in a parametric ways, but we do it for each data point, for each sample from our sample. We learn a conditional flow, and then our marginal flow will just follow by integrating over all of our samples. So these are also sort of the key ideas behind this flow matching that make it distinct from just diffusion models. So we have this conditional objective, which is just for one data point, and we have this objective, which is for the entire samples. And remarkably, you can prove it as a few lines that while these two losses are not the same, they differ by a constant, their gradient are the same. So even if you want to train by this loss, you can in fact train using this conditional loss. That's the key idea. And this simplifies the computation. All right, so I will not go into the details of how to train that. I just wanted to finish with one application of flow matching. Like you've seen, it's a much newer method, so papers in general and in CombBio are just, I think, starting to emerge. So one line of method, which is the first bullet here, is basically similar to RF diffusion. So we're trying to do protein design, but instead of diffusion, you're using flow. So it can be maybe faster, slightly less accurate, but it's trying to solve the same problem. Another interesting paper from last year, it tries to do something else, which is trying to do what's called ensemble modeling, right? So in the protein structure world, there is sort of this basic baseline hypothesis saying that sequence determines structure, right? Which is probably approximately or maybe even highly true for many proteins, but not always true. So sometimes we have proteins or the same molecule, same sequence, but it can have some diversity in its structure, right? And the very well-known method like AlphaFold were trained as regression methods. So they are trained to predict a single structure. So they are not optimal in trying to predict the ensemble of possible structure for the same molecule. And many people are trying in the last few years to tool this method or develop new methods to predict ensembles of structures. So for example, if you have a certain molecule, you can say, okay, it has maybe 2 or 3 stable structure, or maybe it has some variability in part of the molecule, etc. So there are what's called MSA subsampling approaches, and you can think of them as sort of prompt engineering approaches. So AlphaFold is or other methods are big deep learning models, and one of the input to them is the multiple sequence alignment, right? So if you perturb your alignment, for example, by sampling or by clustering, so you basically give the same trained model a different prompt. And sometimes you're lucky and give it a different prompt will give you a different predictive structure, right? So this works sometimes, but only in a few cases. And on the other hand, you have this method, which are called molecular dynamics method. So these are very highly accurate methods. They're actually modeling the physical forces between all atoms in a molecule, but they are very slow. And what you see here is that, so here we see superimposed many samples from this method. So you can see that they are wider. It means that there is some heterogeneity here. So you have this structural motifs, these are called alpha helices. Here you see really just one molecule, but here we have many molecules that you have some flexibility in the protein. And this method from last year was using flow matching together with AlphaFold to actually recover quite well, at least in some cases, the diversity that you observe in molecular dynamics, but in a much faster way. So this is actually computationally tractable. So we see here another application of flow matching for predicting ensembles of proteins. And this is just one example, and you can see here like a more systematic benchmarking. So you can benchmark this method by precision, how well they predict the original structure, and diversity. So do they predict the ensembles? And you see this particular method is here. All right, so let me just wrap up. So we've seen 3 methods for generative modeling: variational autoencoder, diffusion, and flow matching. And these are sort of just the high-level properties of them. How do you train them? How do you sample? So you see this drawback of diffusion models that you need to do many steps. And the quality, so variational autoencoders do not reach the same high quality of samples as the more modern methods, but they are fast, and diffusion models reach higher quality, but they are much harder to train computationally. And flow matching, at least in some applications that it was tried, it reaches almost as high accuracy as diffusion, but in a much lower computational burden. All right, so that's sort of, as you can see, most of these papers are really for the last 2, 3, 4 years, so these things are moving very fast. But this is sort of trying to get a snapshot of the current landscape of this method. All right, so I think I'm done, and I'd be happy to take questions. Thank you.